In this lecture, we're going to look at the Rhabdoviridae, Paramyxoviridae, and Filoviridae families. These are all monopartite negative sense RNA viruses. So let's go back and review this slide that we looked at a while back. Now, all the virus families that we've talked about so far have been positive sense RNA viruses, and they all have one thing in common. The first step that's going to occur when the virus is uncoated in the host cell is that non-structural proteins are going to be translated, specifically polymerase proteins. With minus sense RNA viruses, that can't happen because it's minus sense. It's the wrong polarity to be directly translated into protein. So you have to go through a transcription stage, which requires a specific virus transcriptase, the RDRP, or RNA-dependent RNA polymerase. And that has to be carried in the virion. So when the virion infects a host cell, it carries with it not only the genome that has all the genetic information, but also the transcriptase. And then messenger RNA will be transcribed, which can lead to translation of viral proteins. And then you can have further transcription for replication occurring once you've made more of the RDRP. Rhabdovirus virions are 70 nanometers in diameter and 170 nanometers long, although some are longer and some are shorter. And it consists of an envelope with large peplimers or spikes within which a helically coiled cylindrical nucleocapsid resides. The precise cylindrical form of the nucleocapsid gives rise to the distinctive bullet or conical morphology of virus particles, which you can see in the electron microscopy illustration in this slide. The genome is a single molecule of linear, negative sense, single-stranded RNA. Rhabdoviruses generally have 5 proteins. L, the RNA-dependent RNA polymerase, which functions in transcription and RNA replication. G, the glycoprotein, which forms trimers that make up the peplimers or spikes. N, the nucleoprotein, the major component of the viral nucleocapsid. P, a component of the viral polymerase. And M, a matrix protein that facilitates virion budding by binding to the nucleocapsid and to the cytoplasmic domain of the glycoprotein. 3 proteins, N, P, and L, in association with the viral RNA, constitute the nucleocapsid. The nucleocapsid consists of the genome, bound by the nucleoprotein, N, a phosphoprotein, P, and the large L protein, which is the RDRP. The matrix protein is associated with the inner side of the envelope. The glycosylated envelope protein assembles into homotrimers to form the protein spikes extending from the outer surface of the envelope. Rhabdoviruses appear to use a variety of different receptors for attachment to different types of host cells. Receptors have been difficult to identify because of the generally broad host range and the binding properties of rhabdoparticles. Attachment and penetration are mediated by the surface glycoprotein G. G homotrimers form spikes on the surface of the virion. G is an uncleaved, membrane-anchored protein. Attachment leads to endocytosis, and within endosomes, low pH causes a conformational change to expose hydrophobic loops that participate in fusion. Fusion releases the ribonucleoprotein particle, or RNP, into the cytosol, the site of transcription and genome replication. Due to its role in attachment and penetration, G is an important target of the protective immune responses. For instance, vaccines targeting the rabies virus G protein are highly effective. Following fusion of the virus envelope with endosome membranes, which releases the internal virion components into the cytoplasm of the host cell, the viral M protein dissociates from the nucleocapsid. This step is necessary for viral RNA synthesis to occur because M protein inhibits viral transcription. Remember that we're going to have transcription of messenger RNA as the first step after the virion contents have been released into the cytosol. So remember that along with the genome, you also have your LP polymerase complex, or the RDRP, that was carried in the virion. Transcription begins at the 3' end of the viral RNA, and each of the 5 messenger RNAs are transcribed in order. And you have a poly A tail added at the 3' end before moving on. So there are 5 distinct messenger RNAs for the typical rhabdovirus. You have a 5' cap and a poly A tail. And the various categories of messenger RNA are made in the same proportion as the proteins found in the infected cell, which would be the most abundant protein found in an infected cell is the N protein, which makes sense. The one that occurs in the lowest concentration is the L protein, which also makes sense. Remember, that's an enzyme that can be reused. Transcription begins only at the 3' end of the negative sense RNA. You have sequential transcription. The viral mRNAs are capped and polyadenylated by polymerase stuttering in the cytoplasm. Transcription attenuation is a general feature of non-segmented negative strand RNA viruses. The term attenuation is used to describe the process by which the abundance of transcripts is regulated by their relative proximity to the 3' end of the genome. N messenger RNA is the most abundant transcript, and L is the least abundant transcript, as I mentioned with the previous slide. And you'll notice that that correlates with the order of these genes. The process of attenuation occurs when L completes a transcript and then disassociates from the genome before starting transcription of the next downstream gene. When we look at the transcription of messenger RNA, the first thing that's transcribed from that negative sense RNA genome that comes in when the virus infects a cell is a 48 base leader sequence. This is released and transcription continues to produce the 5 messenger RNAs sequentially. There's a poly A tail that's added at the end of each messenger before the polymerase moves on to the next gene, and this is done through sort of a stuttering mechanism. The result of this is that there's about a 30% chance of the transcriptase falling off the template between transcripts. Each messenger RNA is about 30% less abundant than the predecessor. This attenuation appears to be important for the regulation of the virus life cycle, so that the mRNAs for proteins needed in most abundance are produced in greatest abundance. In the laboratory, studies have been done where the genome has been modified to change the order of the genes. and the resulting modified virus can give rise to viable virus progeny, but the yield is reduced. This demonstrates that the order of the genes as it appears in the normal virus provides a substantial benefit to the replication of the virus. Eventually, there has to be a shift from making messenger RNA to transcribing full-length positive sense RNA. You need to make more full-length RNA in order to produce the genome for the virion, and you have to go through a full-length positive strand intermediate in order to do that. And it turns out that the control of the shift from messenger RNA transcription to full-length positive sense RNA transcription is the concentration of the N protein. The original input RNA that comes in with the infecting virus particle is complexed with N. This is not released for transcription. In the initial stages of infection, N is translated from messenger RNA and concentration increases. Remember, N is the most abundant protein that's produced in an infected cell. When the concentration of N increases, it complexes with the leader sequence as it is transcribed. The leader has a binding initiation site for RNA. This prevents its release, and then transcription continues down, and you get a full-length transcript produced. The messenger RNAs don't bind N because they don't have a binding initiation site. Once the leader is released, the transcriptase is committed to making messenger RNA production. So it's a nicely balanced system. When the virus first infects a cell, you need to make a lot of viral proteins. So the emphasis is on transcription of messenger RNA that can then be translated into viral proteins. The most abundant viral protein that's produced is the N protein. As its concentration rises, then that will cause the shift by binding with the leader sequence, so that rather than being released, as we saw with transcription of messenger, it's retained, and then transcription continues down the full length of the template. Then for assembly and release, the G protein is processed in the ER and the Golgi. It's carried to the plasma membrane in a vesicle. And as is typical for envelope proteins like that, it has a cytoplasmic domain. The M protein, the matrix protein, associates with that cytoplasmic domain of the G protein. And then the nucleocapsid will bind with that, and you have budding through the plasma membrane. Now let's look at the diseases caused by rhabdoviruses. First, we have vesicular stomatitis virus, or VSV. VSV affects hoofed livestock in North and South America. Infections are usually mild, but outbreaks can have a significant economic impact. VSV infection causes formation of blister-like lesions of the mouth, tongue, teats, and hoofs. Outbreaks are sporadic and rare in the U.S. Hosts of the virus in the U.S. include horses, cattle, swine, deer, and occasionally humans do get infected. VSV has been isolated from biting insects, and this is probably how the virus is introduced into a herd. However, once an animal has been infected, transmission can continue as a result of contact. Human infections are generally associated with exposure to infected animals, and in humans, disease is usually a febrile illness. In rural areas where it's endemic, greater than 25% of farmers are seropositive. Human infections are asymptomatic or mild and can be accompanied by a herpes-like lesion in the mouth or nose. But we want to focus on rabies, which is a uniformly fatal disease of mammals. Human rabies is present in 150 countries and territories on all continents except for Antarctica. Tens of thousands of people die each year from rabies. Globally, estimates indicate that human mortality due to endemic canine-mediated rabies is highest in Asia, with the highest incidence and deaths reported in India. This is closely followed by Africa. However, estimates of burden have always been uncertain due to the absence of reliable data. So over 95% of human rabies cases occur in Africa and Asia, mostly impacting children. It's transmitted to people from animals, with 99% of the cases in these countries being caused by dog bites. Rabies is a neglected disease found in poor and disadvantaged populations who often have limited access to health care. Now we have a different situation when we look at rabies in the United States. Outbreaks of rabies infections in terrestrial mammals like raccoons, skunks, foxes, and coyotes are found in broad geographic regions across the United States. Geographic boundaries of currently recognized reservoirs for rabies in terrestrial mammals are shown on the map on this slide. Rabies infections of terrestrial animals occur in geographically definable regions where virus transmission is primarily between members of the same species. Spillover infection to other species occurs, but rarely initiates sustained transmission in other species. Rabies variants can be identified antigenetically by monoclonal antibodies or by genetic analysis. Spatial boundaries of enzootic rabies in reservoir species are temporally dynamic. Affected areas may expand and contract through virus transmission and population interactions. Population increases and emigration result in the expansion of rabies-infected areas, whereas natural barriers, such as mountain ranges and bodies of water, may restrict animal movements or sustain lower population densities. The pattern of rabid animals reported in the United States has changed significantly over the past several decades. Starting in 1938, which is where this graph begins, most of the cases of rabies were in domestic animals, primarily dogs. Now this picture, I like to use this picture in the rabies lecture, but that's actually my pug Riley wrestling with his cousin Lily. He's not rabid, but he does look fierce. But at any rate, we've seen a major shift in the pattern of rabid animals in the U.S., and this started occurring in the 1950s. And it's directly attributable to the development of effective vaccines for dogs and cats. So in 1938, most cases of rabid animals reported were domestic, and some were wild. Since then, the numbers have reversed. In 2017, there were 400 cases of rabid animals reported that were domestic, and 4,000 cases that were from wild animals. The number of rabies cases reported in cats is routinely 3 to 4 times that of rabies reported in cattle or dogs. Wild animals account for more than 95% of reported cases. Raccoons continue to be the most frequently reported rabid wildlife species, followed by bats, skunks, foxes, and other wild animals. Although in 2018, bats actually beat out raccoons, and they were the most frequently reported rabid animal in the United States. However, although only around 30% or so of the rabid animals that are reported are bats, about 90% of human rabies cases in the United States, which is a small number, it's about 1 or 2 per year, originate from contact with bats. Some cases involve sleeping people who didn't know they were bitten, according to the CDC. There is little evidence for human rabies cases involving sleeping adults who did not know they were bitten. There's a big difference between a case of human rabies in which the victim is too sick to report how he or she was exposed, and one who is not aware of being bitten. However, children, some mentally challenged adults, and those with other issues, such as the use of alcohol or medication, are a different matter. Finding a bat in a room with a sleeping child is always a cause for capturing and testing the bat. If that is not possible, the possibility of post-exposure vaccination should be discussed with a medical professional immediately. Vampire bats feed at night and prefer moonless nights to avoid detection by prey animals. The common vampire bat has a number of adaptations for blood feeding that enhance its ability to transmit rabies virus. First, the species feeds on a wide range of hosts, including humans, although will preferentially feed on large livestock such as cattle and horses. The teeth of vampire bats are blade-like and undergo thygosis, a process of self-sharpening in which the upper incisors brush against the lower canines. This ensures that the bat can deliver a virtually painless bite, creating a distinctive crater-like wound on the host. The wound is sufficiently deep to induce profuse bleeding. Clotting is prevented by secretion of anticoagulant in saliva that is channeled down a groove on the dorsal surface of the tongue. Finally, the species forms colonies, occasionally consisting of thousands of animals. It has been conjectured that the European conquest of the New World had a major impact on the vampire bat due to the introduction of livestock, which provided a highly accessible target for vampire predation. Vampire bats show a preference for livestock, particularly larger animals such as cattle, horse, and sheep, because they tend to remain active and stationary at night, in contrast to indigenous wildlife. Also, livestock herds tend to remain in the same location, and the vampire bats are capable of relocating the same site over many nights. Reports of both livestock and human cases of rabies are increasing, mainly due to the continuing encroachment of human populations into areas occupied by vampire bats. And there is now strong evidence that the range of vampire bats is also increasing. It is possible that vampire bats could move further north and into the United States, bringing with them the risk of rabies virus transmission to humans and livestock. Fossils of vampire bats have been found in a number of U.S. states from warmer periods in Earth history. This suggests that possible effects of climate change that lead to increases in temperature could enable the vampire bat to move north and introduce another rabies variant into the U.S. wildlife. Distribution of rabies-affected areas in Mexico over the past several decades is illustrated on this map here. And you can see that the distribution has substantially changed and has increased over the past 30 or so years. Transmission of rabies virus usually begins when the infected saliva of a host is passed to an uninfected animal. Various routes of transmission have been documented and include contamination of mucous membranes, aerosol transmission, and even corneal transplantations. The most common mode of rabies virus transmission, though, is through the bite of an infected animal. All canine rabies vaccines licensed in the United States and Canada are inactivated killed vaccines. The use of these vaccines has successfully eliminated dogs as a reservoir for rabies in the U.S. There is a vaccine that is used for wildlife. This is an oral vaccine. The Raboral VRG is an oral rabies vaccine bait that contains an attenuated recombinant vaccinia virus vector vaccine that expresses the rabies virus glycoprotein gene, or the VRG. The Raboral VRG vaccine has been deemed safe in more than 60 different species of animals, including domestic dogs and cats. The liquid vaccine is packaged inside edible baits, which are distributed into wildlife habitats for consumption by target species. Field application of Raboral VRG has contributed to the elimination of wildlife rabies from 3 European countries, Belgium, France, and Luxembourg, and also eliminated the dog-coyote rabies virus variant from the United States. An oral rabies vaccination program in west central Texas has essentially eliminated the gray fox rabies virus variant from Texas, with the last case reported in a cow during 2009. A long-term oral rabies vaccine barrier program in the U.S. using Raboral VRG is preventing substantial geographic expansion of the raccoon rabies virus variant. And you can see that on this map here. The red areas are where they've distributed the Raboral vaccine that's designed to be attractive to the raccoons. The underlying principle of an oral rabies vaccination program is that a sufficient proportion of a target population may be vaccinated to function as an immune buffer to disrupt the rabies transmission cycle. As the percentage of the target population that's immunized increases, the risk of virus transmission decreases as a function of dwindling numbers of susceptible animals in the population. Unlike population reduction, in which removed individuals are ultimately replaced by susceptible animals, ORV may result in the survival of the immune individuals for their lifespan. So again, the idea here is that you look for areas where you have a borderline between geographic regions that are rabies-free and regions where rabies occurs. And you focus on that border area for the distribution of the oral vaccine. And this can be done by trucks, they can do it by helicopter and planes. But it is proven to be quite effective at controlling the spread of rabies in wildlife. An estimated 59,000 people die from rabies worldwide each year, almost all infected by dogs. Malaria, HIV, and tuberculosis take much higher tolls. But the horrible suffering caused by rabies, some patients have convulsions and become aggressive, just like rabid dogs, and the fact that many victims are children have led the WHO and other groups in 2015 to announce a goal to eliminate rabies deaths worldwide by 2030. In theory, nobody should die from rabies. It's one of the few viral diseases where administering a vaccine after exposure can still save your life. Developing countries try to provide enough doses to hospitals and clinics. Unfortunately, supplies often run out. People who can't afford to buy the jabs from private pharmacies or don't get them in time are doomed. Untreated rabies is the deadliest of all diseases, almost always fatal. That's why the global plan calls for cheaper and faster treatment for people. But its long-term bet is on vaccinating domestic dogs, which pose the biggest threat to people in the developing world. It's the only way you're going to be able to eliminate the program. To rid a canine population of the virus, vets aim to immunize at least 70% of dogs for several years. After that, a lower coverage level could suffice to keep the virus at bay. The dog vaccination strategy has stopped rabies as a killer in developed nations, such as the U.S. And here you can see an example where in Sri Lanka, a dog vaccination campaign was very successful. However, poor countries can hardly pay for millions of dogs to be vaccinated, and their governments often have trouble organizing vaccination campaigns across vast rural areas, even if they have the political will. Big donors, for their part, prefer to work on diseases with a higher death toll or believe that reaching enough dogs is too complicated. In fact, dog campaigns are relatively straightforward, but they often face obstacles. The veterinary teams that typically run them don't work together with public health authorities and tend to prioritize cattle and other valuable livestock, which usually die of other diseases. And that's because there hasn't been the connection made between vaccinating a dog and saving human lives. Although the primary mode of transmission of rabies is certainly through the bite of an infected animal, we have seen cases where rabies was transmitted through organ transplantation. Organ donors are checked for a slew of infectious diseases, including HIV, herpes, hepatitis, and cytomegalovirus. They're not routinely screened for rabies. That's partly because the disease is very rare. An average of only 2 rabies deaths are reported in the United States each year, and partly because rabies testing can take weeks to return results. Human organs have to be transplanted very quickly and typically within hours of a donor's death. In 2004, 3 organ recipients died after receiving transplants from a single donor who, it turned out, had rabies. 3 patients in Germany died after receiving various organs and corneal transplants from an infected donor in 2005. And in 2013, there was an individual who died after contracting rabies from an infected donor, although interestingly, it took 18 months before he became ill. One more. Following primary infection, the virus enters an eclipse phase in which it cannot be easily detected within the host. The phase may last for several days or even months. Investigations have shown both direct entry of virus into peripheral nerves at the site of infection and indirect entry after viral replication in non-nervous tissues, for instance in the muscle cells at the site of the bite. After uptake into peripheral nerves, the rabies virus is transported into the central nervous system through sensory and motor nerves at the initial site of infection. Active cerebral infection is followed by passive centrifugal spread of virus to peripheral nerves. This just means that the virus is now passing from the central nervous system back out into the body of the host using the peripheral nerves. Centrifugal spread of virus leads to the invasion of highly innervated tissues such as the salivary glands. During this period of cerebral infection, the classical behavioral changes associated with rabies develop. The first symptoms of rabies can be nonspecific, just flu-like signs such as malaise, fever, or headache that can last for days. There can even be discomfort at the site of the bite, but this progresses within days to symptoms of cerebral dysfunction, such as anxiety, confusion, agitation, which progresses to delirium, abnormal behavior, hallucinations, and insomnia. The acute period of disease typically ends after about a week. Once clinical signs of rabies appear, the disease is nearly always fatal, and treatment is typically supportive. Disease prevention is entirely prophylactic, and we'll talk about that on the next slide. Non-lethal exceptions are extremely rare. The probability that rabies will develop following an infectious bite depends on the location of the bite, the species doing the biting, and the strain of virus. For instance, if you're bitten on your face or head by an animal, there's a much higher probability of developing rabies than if you were bitten on the leg. Also, bat rabies is an emerging problem in North America. It's different from other rabies viruses. It seems to be able to reach the brain much more quickly than other strains of rabies, and it takes less virus to initiate the brain infection. In many cases of bat rabies infection, there's no known route of infection. Although rabies amongst humans is rare in the United States, every year an estimated 18,000 people receive rabies pre-exposure prophylaxis, and an additional 40,000 receive post-exposure prophylaxis. Pre-exposure vaccination is recommended for persons in high-risk groups, such as veterinarians, animal handlers, and certain laboratory workers. Other persons whose activities bring them into frequent contact with potentially rabid animals such as bats, raccoons, skunks, and dogs should also be considered for pre-exposure prophylaxis. In addition, international travelers likely to come in contact with animals in areas of enzootic dog rabies which lack immediate access to appropriate medical care should be considered for pre-exposure prophylaxis. Pre-exposure prophylaxis is given for several reasons. First, although pre-exposure vaccination does not eliminate the need for additional medical attention after rabies exposure, it simplifies therapy by eliminating the need for human rabies immune globulin and decreasing the number of vaccine doses needed, a point of particular importance for persons at high risk of being exposed to rabies in areas where immunizing products may not be available. And it minimizes adverse reactions to multiple doses of vaccine. Second, it may enhance immunity in persons whose post-exposure therapy might be delayed. Finally, it may provide protection to persons with inapparent exposures to rabies. Post-exposure prophylaxis is indicated for persons possibly exposed to a rabid animal. Possible exposures include, of course, animal bites or mucous membrane contamination with infectious tissues or materials. Administration of rabies PEP is a medical urgency, not a medical emergency. So you don't need to rush immediately to the emergency room, but you don't want to delay your treatment either. Researchers also estimated that about 55,000 Americans were treated for potential rabies exposure each year in 2017 and 2018. The average cost of what's known as rabies post-exposure prophylaxis is about $4,000 per person without hospital or wound care fees, equating to an estimated $209 million spent annually on such treatment. The first step in treatment involves two drugs, a rabies vaccine and something called rabies immunoglobulin. The immunoglobulin essentially kicks the immune system into overdrive, staving off the rabies virus until the vaccine begins to take effect. The immunoglobulin is what interferes before the illness can go from the periphery of the body into the central nervous system and eventually into the brain. It's buying you time. What we find is that it's very expensive to be treated for rabies. To make the treatment, drug companies have to recruit donors and then professionally immunize them with the vaccine against rabies. These people then have their blood drawn and purified to make the immunoglobulin. The drug companies pay these people who are human antibody factories. It's a costly process, as you might imagine. 10 people have survived rabies, including one who received pre-exposure immunization and 8 who received 1 or more doses of post-exposure prophylaxis before the onset of rabies. All but one of these individuals had neurological deficits, and 2 died within a few years. There's a procedure called the Milwaukee Protocol, and that was used on one patient. And that individual beat the odds and survived. But we don't really know whether it was that treatment or not. The Milwaukee treatment has been used on other patients afterwards and not had success. So the question is whether this is just a natural tendency for an occasional survivor or whether the therapy added value. So that's still up to discussion. It may be possible that some people may be able to survive naturally. Paramyxoviruses and orthomyxoviruses were originally grouped together as the myxoviruses. Myxo is Greek for mucus, because of the morphological similarity of the virions and the fact that each carry a hemagglutinin and a neuraminidase. However, it was later realized that the viruses of each group differ in such basic properties as genome structure and mode of replication that they were separated into 2 families. The genome of the paramyxovirus is larger than the rhabdoviruses and encodes more proteins. Most contain at least 6 genes. The P gene uses more than one reading frame to encode multiple proteins. The N, P, M, and L genes serve the same functions as their counterparts in the rhabdoviridae. The G of the rhabdoviridae is replaced by 2 glycoproteins, F and HN. The order of the genes is the same, but they're negative sense, single-stranded RNA genome viruses, so they carry a transcriptase in the virion. The nucleocapsid has helical symmetry, and they are enveloped. Here's another look at the virion structure. N is the nucleocapsid protein that associates with genomic RNA and protects the RNA from nuclease digestion. P is the phosphoprotein that binds to the N and L proteins to form part of the RNA polymerase complex. M is the matrix protein that assembles between the envelope and the nucleocapsid core. It organizes and maintains virion structure. F is the fusion protein that projects from the envelope surface as a trimer and mediates cell entry by inducing fusion between the viral envelope and the cell membrane by class I fusion. One of the defining characters of the members of the family Paramyxoviridae is the requirement for a neutral pH for fusogenic activity. HN contains the receptor binding activity of the virus. These possess both hemagglutinin and neuraminidase activity. Neuraminidase removes sialic acid from receptors and viral glycoproteins. It prevents released virus from aggregating with itself or sticking to infected cells. The virus infects through the respiratory system, so this prevents binding with mucus. The large protein, the L protein, is the catalytic subunit of the RNA-dependent RNA polymerase, or RDRP. Measles virus particles bind to the cell surface receptors via the viral glycoprotein, or HN. Once the viral and cell membranes have been brought together by this receptor-ligand interaction, fusion is induced by a second viral glycoprotein called F, and the viral RNA is released into the cell cytoplasm. The N-terminal 20 amino acids of the F protein are highly hydrophobic and form a region called the fusion peptide that inserts into target membranes to initiate fusion. Because F protein-mediated fusion can occur at neutral pH, it must be controlled to ensure that virus particles fuse only with the appropriate cell and to prevent aggregation of newly made virions. The fusion peptide of F is normally hidden, and conformational changes in the protein thrust it towards the cell membrane. These conformational changes in the F protein, which expose the fusion peptide, are thought to occur upon binding of the HN protein to its cellular receptor. Transcription of paramyxovirus genomes follows the same general process that was described for the rhabdoviruses. Transcription begins when the nucleocapsid is released into the cytoplasm. The bulk of the nucleocapsid consists of the N protein, tightly bound to RNA at the ratio of 1 N protein to 6 nucleotides of RNA. The complex of N protein and RNA is quite stable and serves as the substrate for transcription. The active viral transcription complex of proteins, also part of the nucleocapsid, consists of P and L proteins. There are about 50 copies of L in the virion. So again, this is very similar to what we saw when we were looking at the rhabdoviruses. So N also plays an important role here. So here we see the viral polymerase transcribes the genome template to yield messenger RNAs. That's going to be the first thing that happens because this is a negative strand virus. So it has to carry its transcriptase in the virion, and upon entry and uncoating in the cell, the first thing that's going to happen is transcription of messenger RNA. Eventually, when the concentration of the N protein becomes sufficiently high, the transcription will shift to a full-length copy and eventual transcription of more genomic negative strand RNA. So again, this is exactly the same kind of system that we talked about with the rhabdoviruses. Okay, then we have assembly and budding. So the glycoproteins are inserted into the plasma membrane. The RNP will bind with the M protein. Remember, we said the M protein tends to organize the virion. So you have F and HN spanning the envelope, and then M will bind with the cytoplasmic domain of the two envelope proteins, and then the RNP will bind with M, and eventually you get budding of the virus. And then this is the complete paramyxovirity life cycle. So you have binding with fusion, followed by encoding. Remember, the first step is going to be transcription of messenger RNAs. Those will be translated, and eventually, when the concentration of the N protein is sufficient, rather than making more messenger RNA, you're going to make full-length positive antigenome RNAs, which then can be used to replicate more full-length minus sense RNA that can be packaged into virions. You get assembly at the plasma membrane, followed by budding. Okay, there are a number of really important diseases that fall into this family, and we're going to go through each of these separately. I picked the most significant diseases to talk about in detail. Mumps is a contagious disease caused by a paramyxovirus. It spreads through saliva or mucus from the mouth, nose, or throat. An infected person can spread the virus by coughing, sneezing, or talking, also sharing items such as cups or eating utensils, touching objects or surfaces with unwashed hands that are then touched by others, and other similar kinds of mechanisms of transfer can occur. Mumps likely spreads before the salivary glands begin to swell and up to 5 days after the swelling begins. So this young kid in the picture, he looks like he's got a bit of a double chin there. Well, no, that's because his parotid salivary glands are swollen due to virus infection. So this slide looks at the pathogenesis of mumps virus. Remember, mumps is a respiratory virus. That's how it's spread. It causes an acute inflammatory disease in humans, which causes a range of systemic symptoms. The incubation period is 2 to 4 weeks. Approximately 1/3 of infections are asymptomatic. The prodromal phase is characterized by nonspecific, often mild symptoms such as a low-grade fever or headache and malaise. An early acute phase follows, likely representing spread of the virus from the respiratory tract and development of systemic symptoms, typically infection of the salivary glands, which lasts from a few days to a week. During the established acute phase, orchitis, meningitis, or encephalitis may occur. Symptoms usually resolve within 2 weeks, coincident with mumps virus specific humoral responses. Long-term complications and death are rare. Before the U.S. mumps vaccination program started in 1967, about 200,000 cases were reported each year, but the actual number of cases was likely much higher due to underreporting. Since the 2-dose MMR vaccination program was introduced in 1989, U.S. mumps cases decreased more than 99%, with only a few hundred cases reported most years. However, since 2006, there have been several increases in cases and outbreaks about every 5 years. So mumps is a really contagious viral disease. It affects children, adolescents, and adults. And there have been outbreaks of mumps recently in close-knit communities, such as college campuses. The postulated reasons for the outbreaks, which have occurred amongst individuals who have received the recommended 2-dose vaccination regimen, include the potential waning of the vaccine effect and the lack of long-term boosters. 2 doses of the vaccine are 88% effective at preventing mumps. 1 dose is 78% effective. We normally see a couple hundred of cases per year in the U.S., but there have been occasional outbreaks. Mumps outbreaks can occur any time of year, but often occur in winter and spring. A major factor contributing to outbreaks is being in a crowded environment, such as attending the same class, playing on the same sports team, or living in a dormitory with a person who has mumps. The MMR vaccine prevents most, but not all, cases of mumps and complications caused by the disease. Again, as I said, 2 doses of the vaccine bring the protection range up to about 88% effectiveness against protecting. But these outbreaks can still occur even in highly vaccinated U.S. communities, particularly in close contact settings. In recent years, outbreaks have occurred in schools, colleges, and camps. However, high vaccination coverage helps limit the size, duration, and spread of mumps outbreaks. For instance, in 2015-16, outbreaks were reported from several university campuses, including a number of smaller outbreaks with limited spread. The 2 largest outbreaks were from Iowa and Illinois, each involving several hundred university students. Both held wide-scale vaccination campaigns. There was an outbreak of mumps in 2014 amongst NHL players. So those kinds of things can occur. And just this past spring, there was an outbreak resulting in about 30 cases at a ski resort in Colorado. In terms of complications, you can have orchitis, or swelling and infection of the testes, in males who have reached puberty. This is why mumps can occasionally be a major problem for adult males, because when an adult male has orchitis, it can lead to fertility problems. It can cause encephalitis and meningitis in individuals. You can get inflammation of ovaries and breast tissue. There's evidence of it causing pancreatitis, or inflammation of the pancreas. And as a very severe complication, occasionally it can result in deafness. Now let's move on to measles. With measles, infection begins in the upper respiratory tract but becomes systemic. Lymphoid organs and tissues are common sites of virus replication. One consequence of measles infection is immune suppression, and that can then set the patient up to be susceptible to secondary infections that can also be serious. This virus is spread by aerosol. It'll remain latent in the patient for almost 2 weeks. The patient will develop a prodrome of fever and conjunctivitis, followed by a rash. Onset of rash coincides with the appearance of immune response and initiation of virus clearance. There's a very high rate of mortality in immunologically naive populations. It's estimated that 56 million people in the Americas died of this old world disease following the European exploration of the New World. So when the explorers came from Europe, where measles was an endemic disease, they encountered people in the New World who were immunologically naive to this disease, and it was a horrific event with millions of people dying. This is thought to have facilitated the conquest of the Americas by the Spaniards. Subsequent depopulation of Central and South America allowed them to remain dominant. This may have led Europeans to introduce Africans as slaves, to replace the Native Americans that were dying from measles and smallpox. Measles is an acute, highly communicable viral disease with fever, conjunctivitis, cough, and Koplik spots. You can see the spots, the Koplik spots on the lower left figure there. Those are the spots that appear on the palate of an individual when they're infected with measles. So they're small, red, irregularly shaped, and they tend to have little blue-white centers, and you'll find them in the oral cavity. And Koplik spots will show up when the person is infectious, but before the rash shows up. And that's what's important to notice about this figure. The rash doesn't show up until the viremia is decreasing. The rash is actually a result of the immune response, and the rash is not infectious. In the person, once they've developed the rash, they've actually become less infectious. So it's during the prodromal period when you have Koplik spots and conjunctivitis that the person is most infectious. Measles causes infection by fusing with the plasma membranes of host cells in a receptor-dependent manner. When the measles virus enters the respiratory tract, it infects alveolar macrophages in the lungs first. The primary role of these specialized immune cells is to engulf and destroy foreign substances like dust, bacteria, and viral products. Alveolar macrophages also possess a membrane glycoprotein called signaling lymphocytic activation molecule, or SLAM, that has been identified as the high-affinity cellular receptor for measles. Measles uses SLAM to fuse directly with the plasma membrane, bypass destructive phagocytosis, and release its genome and replication machinery directly into the cell cytoplasm. Instead of destroying the measles virus upon contact, these hijacked macrophages transport viral copies straight to the closest lymph nodes for dissemination. Infected macrophages travel to lymph tissue where the virus comes in contact with the memory cells of the immune system. These lymphocytes are recon strategists. They identify foreign invaders through antigen detection and process these molecular patterns to generate long-lived memory cells for future protection. If a second encounter occurs, memory cells will mount a faster and stronger immune response to that pathogen than during the first encounter. And this, of course, is something that we've already discussed. Measles virus has an intrinsic ability to enter both immune and epithelial cells using different receptor binding sites. As I explained in the previous slide, measles virus initiates its infection via SLAM-mediated entry into alveolar macrophages. After initial replication in regional lymph nodes, the virus enters the bloodstream and subsequently spreads to all lymphoid organs, such as the spleen, thymus, appendix, the tonsils, and lymph nodes throughout the body. When the measles virus infection in the lymphoid organs reaches its peak, measles virus infected immune cells transfer the virus to epithelial cells through the basolateral side. Remember that epithelial cells are polarized. At this step, the virus uses a different receptor, Nectin-4. In the epithelia, measles virus selectively releases its progeny virus particles from the apical membrane to the luminal side of the respiratory tract. This will facilitate virus shedding and spread to another host. And this goes back to what we talked about in terms of polarized trafficking of virus in host cells. So it infects through the basolateral side, but it's released through the apical membrane. Measles is highly contagious, and it lives in the nose and throat mucus of an infected person. It can spread to others through coughing and sneezing. Also, measles virus can live for up to 2 hours in an airspace where the infected person coughed or sneezed. If other people breathe the contaminated air or touch the infected surface, then touch their eyes, nose, or mouths, they can become infected. Measles is so contagious that if one person has it, up to 90% of the people close to that person who are not immune will also become infected. Infected people can spread measles to others from 4 days before through 4 days after the rash appears. At one time, the disease was epidemic throughout the world. It was very infectious, so few could avoid it. Following the introduction of a vaccine, the number of cases dropped dramatically. Virus continued to circulate among non-immunized individuals, and thousands of cases continued to occur. There is a 5% failure rate in the attenuated virus vaccine, so that's pretty good. In 1989 to 91, there was an epidemic of about 50,000 cases. This occurred in young immunized adults as well as young children who had not been immunized. Some young adults became infected due to vaccine failure, but also because of waning immunity. That's why the guidelines now call for a second dose of the MMR vaccine. In 1998, there were only 100 cases in the U.S. The virus is still a problem in developing countries. One issue is that the current vaccine is ineffective in the presence of maternal antibodies, which are usually good for about 9 months. So if the vaccine is given to a child at that time, the vaccine won't take. In the U.S., the first inoculation is given at 12 months, followed by a second dose several years later. If virus is pervasive, a kid will become infected almost as soon as maternal antibody wanes, and this helps to keep the virus in circulation in populations. Okay, so what is in the measles vaccine? First of all, it's grown in chick embryo cells, so people with chicken allergies and so on would have to avoid it. There are stabilizers such as sugar, salt, gelatin, and an antibiotic, neomycin sulfate, is added. The vaccine has to be kept between 2 and 8 degrees centigrade from the time it leaves the warehouse until it's administered. This can be a challenge in developing countries, particularly in tropical environments where power outages are frequent. Health workers have to be trained to inject it safely, and this is a much more difficult task than just dropping liquid polio vaccine on a child's tongue. The vaccine comes as a powder that has to be reconstituted with an accompanying solution of sterile diluent and then used within 6 hours. It also comes in 10-dose vials. Sometimes vaccinators who are worried about wastage will be hesitant to open one when just a couple of kids show up for a session, so the kids may go unimmunized. A new vaccine delivery system is being developed that could be a game changer for measles control and elimination. The vaccine uses a technology called a microarray patch, and you can see a picture of it on the right side of the slide. It looks like a small round bandage with hundreds of microneedles, each carrying a small amount of live, freeze-dried vaccine. It's applied with a small push and delivers vaccine under the skin and within 5 minutes. It's very thermostable, takes up little space, doesn't have to be reconstituted, and you don't have to worry about safety. But unfortunately, the measles patch has languished in development for lack of funding and has yet to reach clinical trials, although these are intended to begin by the end of 2020 or in early 2021. Measles was eliminated in the United States in 2000. High national coverage with measles, mumps, and rubella vaccine and rapid implementation of measles control measures prevent widespread measles transmission. In 2014, the U.S. experienced 23 measles outbreaks, including one large outbreak of almost 400 cases occurring primarily amongst unvaccinated Amish communities in Ohio. Many of the cases in the U.S. in 2014 were associated with cases brought in from the Philippines, which experienced a large measles outbreak. The majority of the people who got measles were unvaccinated. Even though endemic transmission of measles has been eliminated in the U.S., it's still common in many other countries and can be imported by foreign visitors or returning travelers who are not fully protected against the disease. Measles is still common in many parts of the world, including countries in Europe, Asia, the Pacific, and Africa. So travelers to those countries have to be very careful. Measles can spread when it reaches a community in the U.S. where groups of people are unvaccinated. In 2019, 1,200 U.S. measles cases were reported, the highest annual number since 1992. 89% of measles patients were unvaccinated or had an unknown vaccination status, and 10% were hospitalized. 86% of cases were associated with outbreaks in under-immunized, close-knit communities, including 2 outbreaks in New York Orthodox Jewish communities that threatened measles elimination status in the United States. As of October 15, 2020, there have been 12 confirmed cases in 7 jurisdictions here in the U.S. Measles vaccines were introduced 50 years ago and were followed by striking reductions in child morbidity and mortality. Measles control is now recognized as one of the most successful public health interventions ever undertaken. Despite this, in many countries, vaccination targets remain unmet, and measles continues to take hundreds of thousands of lives each year. Even where control has been successful, vaccine hesitancy threatens the gains that have been made. The introduction of mass measles vaccination has reduced childhood mortality by 30 to 50% in resource-poor countries and up to 90% in the most impoverished populations. The observed benefits cannot be explained by the prevention of primary measles infections alone, and it may actually be related to another phenomenon that we'll talk about in a minute. The highly contagious measles virus continues to spread around the globe. In 2018, cases surged to an estimated 10 million worldwide, with 140,000 deaths. This was a 58% increase since 2016. In rich countries, scattered measles outbreaks are fueled by people refusing to vaccinate their children. But in poor countries, the problems are health systems so broken and underfunded that it is nigh on impossible to deliver the vaccine to people who need it. And this situation will only worsen with the COVID-19 pandemic. More than 20 countries have already suspended measles vaccination campaigns as healthcare workers scramble to deal with the coronavirus. If you look at this map, in each of the regions on the map, a slightly different mix of factors led to an outbreak. For instance, in Madagascar, a shortage of measles vaccine helped fuel an outbreak that swept the island nation starting in 2018, causing more than 240 cases and 1,000 deaths. In Ukraine, after a child died of unrelated causes following a measles jab in 2008, vaccination coverage plummeted from 95% that year to a low of 31% in 2016. No one was surprised when, in 2017, a huge outbreak hit that led to more than 115,000 cases. The DRC has difficulties on a number of levels. The country has such a high birth rate, 3.5 million children are born each year, that it needs to conduct mass vaccination campaigns every 2 years. Those campaigns, in which tens of thousands of healthcare workers fan out across this vast country, are a logistical nightmare. First, the government has to get the vaccine from the capital to remote villages that can be reached only by helicopter or through bloody conflict zones in the eastern part of the country. It's just a unfortunate blend of circumstances that make it very difficult for this country. The United States experienced a large multi-state measles outbreak linked to an amusement park in California. The outbreak likely started from a traveler who had become infected overseas with measles, then visited the amusement park while infectious. However, no source was identified. Analysis by CDC showed that the measles virus type in this outbreak was identical to the virus type that caused the large 2014 outbreak in the Philippines. Measles can be serious in all age groups. However, there are several groups that are more likely to suffer from measles complications. People who are at highest risk would be children under 5 years of age, adults older than 20, pregnant women, and of course anyone with a compromised immune system would be at risk. The most common types of complications would be conjunctivitis, inflammation of the voice box, ear infections. Those occur in about 1 out of every 10 children with measles, and it can result in permanent hearing loss. About 5% of children with measles gets pneumonia, and about 1 child in every 1,000 who gets measles will develop encephalitis. This inflammation of the brain can lead to convulsions and can leave the child deaf or with significant mental deficits. For every 1,000 children who get measles, 1 or 2 will die from it. Measles can also make a pregnant woman have a miscarriage, give birth prematurely, or have a low birth weight baby. In developing countries where malnutrition and vitamin A deficiency are common, measles has been known to kill as many as 1 out of 4 people. It is the leading cause of blindness among African children. Measles kills almost 1 million children in the world each year. Okay, there's an interesting phenomenon that occurs after a measles infection called immune amnesia. Now, you remember we said earlier that the measles virus infects a host cell by attaching to the SLAM surface receptors. Well, it turns out that memory T cells and B cells also contain SLAM surface receptors. Research has shown that measles virus binds and infects these memory cells and also naive B cells of the immune system. Once infection is established, the virus spreads through the body by budding from infected cells. Clearance of measles virus requires the elimination of virally infected lymphocytes. Immune-mediated destruction of memory T cells and B cells is initiated, and memories of past infections are destroyed along with them. The number of T cells and B cells significantly decreases during the acute stage of measles infection, but there is a rapid return to normal white blood cell levels after the virus is cleared from the system. This observation masked what was really going on until researchers were able to evaluate the qualitative composition of required lymphocyte populations. We now know that the memory T cells and B cells that are produced immediately following infection are dramatically different from those that existed before the measles infection. Not only have pre-existing immune memory cells been erased, but there has been a massive production of new lymphocytes. And these have only one memory: measles. Thus, the host is left totally immune to measles and significantly vulnerable to all other secondary infections. It takes approximately 2 to 3 years post-measles infection for protective immune memory to be restored. If measles infection essentially resets a child's developing immunity to that of a newborn, revaccination or exposure to all previously encountered microbes will be required in order to rebuild proper immune function. That means infections other than measles can result in death due to the effect of measles on the immune system. Because the virus attacks the immune system, the severity of infections depends on the general health of the population. In essence, measles acts like an infection amplifier, turning up the volume of background disease. This means that in resource-limited areas, the virus will cause higher overall death rates. In lots of developing countries still today, we see 1 in 50 or 1 in 100 kids dying from measles, primarily due to these immune effects. Distemper is a highly contagious disease and fatal about 50% of the time, thus making it the leading cause of infectious disease death in dogs. The virus infects the gastrointestinal tract, the respiratory tract, the brain, and the spinal cord. Common symptoms can include high fever, watery discharge from the eyes and nose, vomiting and diarrhea, hardening of the foot pads and nose, seizures, and paralysis. It is commonly associated with domestic animals such as dogs and ferrets, although it can infect wild animals such as raccoons. Most often puppies, but all ages are susceptible. No treatment is available, just supportive care. Convalescence can be long. Infected animals should be quarantined from other dogs for several months due to the length of time the animal may shed the virus. Prevention through vaccination is the best control. Vets give the vaccine at 6 weeks of age and continue at 3 to 4 week intervals until the dog is about 16 weeks old. Antibodies in the mother's milk can interfere with the vaccine, so that's why we have to give it multiple times. We find that the antibodies in the mother's milk interferes about 75% of the time when the puppy is 6 weeks old. This drops down to 25% at 9 weeks, and at 12 weeks, very few are showing any kind of interference from the mother's milk. Some puppies develop signs of distemper following vaccination, although they don't appear to have the disease. The prevalence of canine distemper in the community has decreased dramatically due to the availability of vaccinations. However, the disease continues to spread among unvaccinated populations, such as at animal shelters and pet stores. Because it likes to infect and damage epithelial cells, canine distemper also tends to infect the outer skin of the dog. This damage results in a number of different kinds of skin lesions, the most frequently being the hard pads on the feet and the hard nose, which can be a real distinctive sign. Nipah virus is a paramyxovirus whose reservoir host are fruit bats. Occasionally, the virus is introduced into human populations and causes severe illness characterized by encephalitis or respiratory disease. Zoonotic transmission cycles in Nipah virus in Malaysia and Bangladesh have been seen. The fruit bats are the natural reservoir, and it's transmitted from bats roosting in fruit trees on pig farms to pigs, and subsequently pigs transmit the Nipah virus to people in close contact with those pigs. In some areas, Nipah virus is thought to be transmitted by the consumption of raw date palm sap. While date palm sap is collected, bats drink from the sap stream or collection pots and contaminate the sap with Nipah virus through their saliva or urine. People become infected with Nipah virus after drinking this contaminated date palm sap. Subsequently, these infected people can transmit Nipah virus to others via close contact. Respiratory syncytial virus, or RSV, is the most common single cause of respiratory hospitalization of infants and is the second largest cause of lower respiratory infection mortality worldwide. In adults, RSV is an under-recognized cause of deterioration in health, particularly in frail elderly persons. Infection rates typically rise in the late autumn and early winter, causing bronchiolitis in infants, common colds in adults, and insidious respiratory illness in the elderly. For 2013 to 2014, the RSV season onset ranged from late October to late January, and season offset ranged from late January to early April in all 10 Department of Health and Human Services regions, except Florida. Florida has an earlier RSV season onset and a longer duration than the rest of the country. Now let's look at some of the symptoms that kids can develop. First, we have croup. That's a viral condition that can be caused by a lot of different viruses, including RSV. So croup is just a symptom, but it's characterized by breathing difficulties and a bad cough that can sound like a barking seal. Another problem that can occur from a number of viruses, including RSV, is bronchiolitis. With this, you get an edema of the bronchiolar walls, necrosis of epithelium, you get necrotic material that blocks the lumens of airways, and you get an increased secretion of mucus. This is especially dangerous for infants because their bronchioles are very narrow and prone to obstruction. Resistance to expiration can lead to hyperinflation of the lungs. In other words, because these airways are so narrow, when they try to breathe out, they can't, and the lungs slowly become overinflated. But you can hear a characteristic kind of wheezing sound from these children. And then there's always pneumonia, which of course has many possible causes, including RSV. You can diagnose pneumonia using X-ray, but also a doctor will use lung osculation. That's when they go in with the stethoscope and listen to you breathing. They'll tell you to breathe in and out, and they're listening for crackles. And that's a diagnostic sign that the patient has pneumonia. Now, what happens with infection with RSV is that the G protein binds to its receptor on the surface of ciliated epithelial cells in the mucous membranes of the nose and throat. Once the virus and cell membranes are in contact, fusion is induced by the F protein, and the nucleocapsid is released into the cytoplasm. Progeny from the first infected cell can spread to other cells by shedding from the apical surface. However, cell-to-cell fusion can occur, resulting in multinucleated giant cells, or syncytia. That's where it gets its name, and you can see on the right-hand side of this illustration the syncytia that have formed in infected cells. So this is basically what's happening. You have the virus-infected cells that are expressing viral proteins, viral envelope proteins, on their plasma membrane. Those viral proteins can bind with the receptors on an uninfected cell, and the infected cell and the uninfected cell can fuse. And this can keep repeating until you get the formation of large syncytia. Now, the last virus I want to talk about in this lecture would be the filoviridae. The filoviridae is a family of viruses that contain single, linear, negative-sense RNA genomes. The family name was derived from the Latin word phylum, which alludes to the thread-like appearance of the virions when viewed under an electron microscope. The virions are very long and thread-like, and they take on odd shapes like shepherd's crooks and so on. They're variable in length, but uniform in diameter. The tubular Ebola virions are usually 80 nanometers in diameter and approximately 800 nanometers long. In the center of the particle is the viral nucleocapsid, which consists of the helical single-stranded RNA genome wrapped about the NP, VP35, VP30, and L proteins. The structure is then surrounded by an outer viral envelope derived from the host cell membrane that is studded with 10 nanometer long viral glycoproteins, or GP spikes. Between the capsid and envelope are viral proteins VP40 and VP24. The envelope GP spike is responsible for viral attachment and membrane fusion. Virus particles bind to a cell surface receptor followed by uptake via macropinocytosis and trafficking to late endosomes. GP is cleaved by endosomal cysteine proteases and then binds to the membrane protein NPC1, which is exposed in the endosomal lumen. The interaction leads to fusion of the viral and endosomal membranes and delivery of the viral nucleocapsid into the cytoplasm. The central core of each filovirus virion consists of the single RNA genomic molecule and its adherent nucleoproteins, NP and VP30. This forms the nucleocapsid, or NC. They are linked by matrix proteins, VP24 and VP40, to the inner surface of the lipid bilayer of the viral envelope, which is derived from the host cell. Trimeric spikes of the surface envelope glycoprotein, or GP, are anchored in the bilayer. Two other proteins, VP35 and the large protein, the L protein, which is the RDRP, are also present within the virion. Just as in other negative-sense RNA viruses that we've talked about already, the transcription process begins with the binding of the polymerase complex to a single binding site located within the leader region of the genome. The complex then slides along the RNA template and sequentially transcribes the individual genes in their 3' to 5' order. However, the polymerase is released from the template following mRNA formation, so reinitiation at downstream genes is attenuated. Thus, the first gene, NP, is transcribed at the highest levels, whereas the last gene, L, is transcribed at the lowest. So attenuation of the transcription of messenger RNAs, just as we talked about with the rhabdoviruses and the paramyxoviruses. The concentrations of viral proteins, especially NP, regulate the switch from mRNA synthesis to genome replication, which begins with synthesis of full-length positive strand copies of the viral RNA. These are encapsulated by NP and in turn serve as templates for the synthesis of full-length minus strand RNAs. Again, very similar to what we've already talked about. Inclusion bodies are the sites of RNA synthesis and nucleocapsid assembly. Assembly begins with the synthesis of GP in the ER and transport to lipid raft domains at the plasma membrane. Octamers of VP40 are produced and transported to the GP-containing rafts. VP40 interacts with the C-terminus of NP and directs viral minus-strand nucleocapsids to sites of virus budding. The nucleocapsids form parallel to the plasma membrane, and virus particles are released by budding. So the interaction of nucleoprotein NP with the matrix protein VP40 is essential for the transport of nucleocapsids to the cell surface and for the incorporation of these NCs into virions to form mature virus particles. A unique aspect of Ebola virus budding is that the particles containing NCs mainly emerge horizontally from the cell surface. Most other viruses bud vertically. In terms of classification of the Filoviridae, there are two genera, the Ebola virus and the Marburg virus. And we'll talk about these separately as we go along. So let's first talk about Ebola. There are 5 distinct species of Ebola virus. The Bundibugyo, Sudan, and Zaire species have been associated with large outbreaks of Ebola hemorrhagic fever in Africa with high mortality, while Cote d'Ivoire and Reston have not. The Reston Ebola virus has origins in the Philippines. It was first discovered in a quarantine facility for cynomolgus monkeys in Reston, Virginia, and this is the subject of the book you've been assigned to read, The Hot Zone. Luckily for the monkey caretakers, this is the only strain that is non-pathogenic in humans. Recently, Reston Ebola virus has been found infecting pigs in the Philippines. This raises important concerns for the public health and food safety in this country, causing some to worry that this could turn into a serious problem for parts of Asia. Ebola hemorrhagic fever is thought to be a classic zoonosis with persistence of the Ebola virus in a reservoir species that is found in endemic areas. Apes, man, and other species that are susceptible to Ebola virus infection are regarded as dead-end hosts and not as reservoir species. Ebola is introduced into the human population through close contact with the blood, secretion, organs, or other bodily fluids of infected people or animals. In Africa, infection has been documented through the handling of infected chimpanzees, gorillas, fruit bats, monkeys, forest antelope, and porcupines found dead or ill in the rainforest. In Africa, fruit bats are considered possible natural hosts for Ebola virus. As a result, the geographic distribution of Ebola virus may overlap with the range of the fruit bats. The Ebola virus enters the host primarily through mucosal surfaces or skin abrasions, with most human infections occurring after direct contact with patients or cadavers. How you get infected also plays a role in how sick you become. An exposure that delivers the virus directly into the bloodstream, for example, a needle stick injury, is more damaging than when the viruses are introduced by the mucous membrane surrounding the eyes, nose, and mouth. Onset of symptoms can be as little as 2 days with direct-to-blood exposures, while the incubation period can range up to 21 days for mucosal or broken skin entry. Most infections become apparent within 8 to 10 days of exposure. In addition, there have been several cases in which hunters are infected after eating contaminated meat found in the forest. Also, most Ebola outbreaks are centered in hospitals in Africa, where practices of basic hygiene and sanitation are lacking. However, in modern hospitals with disposable needles and knowledge of basic hygiene and barrier nursing techniques, the virus rarely spreads on a large scale. Airborne transmission between monkeys was demonstrated during the outbreak of Reston Ebola virus in Virginia, but there's limited evidence of airborne transmission in any human epidemics. The severe illness results from a complex of pathogenic mechanisms that enable the virus to suppress innate and adaptive immune responses, infect and kill a broad variety of cell types, and elicit strong inflammatory responses in disseminated intravascular coagulation, producing a syndrome resembling septic shock. So the virus enters through contact. It starts to destroy white blood cells. Infected cells carry the virus throughout the body. It can lead to the formation of blood clots that will cause very severe organ damage. The immune system goes into a dangerous overdrive known as a cytokine storm. So essentially, the immune system is turning against itself. The disease can attack any organ in the body, including the brain, the liver, the eyes, the genitals, the intestines. The vascular system is severely damaged. You get internal and external bleeding from wounds, mucous membranes, and orifices. And then eventually death can be caused by organ failure or by the patient going into shock from the loss of fluid or blood supply. One disturbing aspect of the Ebola virus is that even if it doesn't kill you, it can cause lasting problems, such as joint pain, fatigue, mental issues, and visual changes. By some measures, 20 to 25% of survivors at some point experience a dangerous inflammation within the eye, and around 40% of those who experience this eye condition go on to become blind or have severe visual impairments. So this is an interesting illustration here. What you see is a picture of the eyes of an American physician named Ian Crozier, who contracted the virus in West Africa during the outbreak in 2014. He survived, but months after the acute infection passed, he reported visual problems, and physicians discovered that the Ebola virus had made its way into his eyes. He had severe inflammation and dangerously high eye pressure. Surprisingly, his left eye turned from blue to green, and then back to blue after physicians treated him with an experimental antiviral drug. He's now regained normal vision. Ebola was first identified in 1976 and occurs primarily in regions of sub-Saharan Africa. There are normally fewer than 500 cases reported each year. and no cases were reported between 1979 and 1994. But you can see on this diagram the very high bars that indicate the outbreak that occurred in 2014. The WHO confirmed an outbreak of Ebola virus disease in southeastern Guinea in March 2014. This was the first time an outbreak had been diagnosed in this part of Africa, and the index case was traced back to December 2013. The outbreak spread to neighboring and nearby countries, particularly Liberia and Sierra Leone, and became the largest ever known outbreak of Ebola. It continued for more than 2 years, and a total of 28,616 confirmed, probable, and suspected cases were reported in Guinea, Liberia, and Sierra Leone together, with over 11,000 deaths. This shows you the cases of Ebola that occurred outside of Africa during this outbreak. In all but 3 cases, the patient was infected with Ebola while in West Africa, and then the disease was imported to these other countries. Infection outside Africa has been restricted to health workers in Madrid and Dallas. For instance, in the United States, there was a single Ebola case that was imported from Liberia to Texas in September 2014. This resulted in transmission to 2 healthcare workers who were working in the hospital caring for this patient. A further case of Ebola was confirmed in a humanitarian aid worker who returned from Guinea in October 2014, but that was an imported case. So in the U.S. during this 2014 outbreak, we did have 2 cases of Ebola transmission to healthcare workers. Just when health officials would think that the Ebola outbreak was over in West Africa, the virus would pop up again, seemingly out of the blue. It happened several times during the 2013 to 2016 outbreak. Now, scientists are starting to figure out why. The virus can lie dormant in a survivor for more than a year and reemerge to infect others. So, in other words, Ebola is capable of causing a persistent infection in infected individuals. It's rare, but it's played a big role in keeping Ebola around in Liberia. We believe that most, if not all, of the new clusters of Ebola cases that popped up were from persistent infections in survivors, but it can be hard to determine that for sure. But there are a number of areas in the body that are immune privileged where Ebola can hide out. We just saw that that can occur in the eye, but it can also be in breast milk, the central nervous system, and testes. Research on where, how long, and at what concentration the virus can survive is ongoing. But if the virus is hiding out in an infected individual in a persistent infection in immune privileged pockets, there's always that possibility that the virus could break out of these privileged areas, establish an active infection in the patient, and possibly trigger a new outbreak. An Ebola glycoprotein mutant that arose early during the West African epidemic increased the infectivity of the virus to human cells and may have contributed to increased mortality. The magnitude of this West African Ebola epidemic enabled an unprecedented number of viral mutations to occur over successive human-to-human transmission events, increasing the probability that adaptation to the human host could occur during the outbreak. One set of investigators looked at a mutation in the GP or glycoprotein for its effect on viral infectivity. This mutation, it's indicated by V in the graph here, it heightens the ability of the virus to infect primate cells, including human dendritic cells. It's located at the NPC1 binding site on GP. And what we saw is that during the outbreak, it rose to very high frequency. The increased infectivity that was seen was restricted to cells that have primate-specific NPC1 sequences at the Ebola virus interface. This suggests that this mutation was indeed an adaptation to the human host. The new genotype was associated with increased mortality, which is consistent with the hypothesis that the heightened intrinsic infectivity of this genotype contributed to the disease severity during the West African epidemic. In August 2018, a new epidemic in Africa of Ebola was declared. This was in the Democratic Republic of the Congo, or the DRC. By the time it was declared over in 2020, it had infected at least 3,470 people, killing 66% of them. That made it the world's second largest outbreak of Ebola after the 2014 to 2016 West African epidemic, which killed more than 11,000 people. Experts say that the northeastern epidemic, which infected mostly North Kivu and Ituri provinces, and you can see that on the map on this slide, this was one of the most complex health emergencies that healthcare workers around the world have had to deal with, because it occurred in a region of the DRC that's been plagued by 25 years of war and political instability. In the course of the outbreak, more than 70 Ebola patients and Ebola responders were injured in dozens of targeted attacks by armed groups. At least 11 were killed. However, the epidemic was marked by successes in vaccination and treatment. This was the first Ebola outbreak in which a vaccine for the virus was widely deployed. The vaccine, made by the drug company Merck, and was first tested during the West African epidemic, was given to more than 300,000 people who had been in close proximity to people with Ebola and their contacts. More than 80% of the people who were vaccinated didn't end up with the disease, and those that did had relatively mild cases, so very good results. A clinical trial conducted during the outbreak also found that 2 antibody-based drugs reduced deaths dramatically among people who were hospitalized soon after being infected. These were then given to all consenting patients in Ebola treatment centers in the northeastern DRC. Another key success was local leadership. The rate of new infections in the region slowed after the governors and mayors of the cities hit hard by Ebola began pushing to stamp out the virus. And once local health workers had been trained and equipped to care for people, a lot of successes were seen. So what happened here is that lessons that were learned during the West African outbreak and new treatment possibilities, including a vaccine and 2 antibody-based drugs, were deployed very early, and as a result, the epidemic was brought under control. On the 1st of June 2020, the DRC declared yet another Ebola outbreak. This occurred in the Equator province in northwestern DRC, so the opposite side of the country from where the the last outbreak occurred. There does not appear to be a link between this outbreak and the one that occurred on the other side of the country. Fortunately, the response to this new Ebola epidemic does not face the same kind of security challenges that the Kivu and Ituri provinces presented because this part of the country is more stable politically. The outbreak has proceeded slowly and seems to be under control. As of November 1, no new cases have been reported for more than 30 days. However, new clusters of cases can still occur even after long periods of no new cases being reported. The patients in this new outbreak are benefiting from the scientific progress that's been made in recent years, with the vaccine and curative treatments now available. So, for instance, 40,000 people in this outbreak zone were vaccinated using the VSV-ZBOV vaccine. So this is really an exciting time right now because there's some very strong evidence to suggest that there are now good treatments for Ebola and a very effective vaccine. So let's look at these in a little more detail. The RVSV-ZBOV vaccine is a recombinant virus in which the VSV envelope glycoprotein of VSV is replaced with the Zaire strain Ebola virus glycoprotein. This vaccine is currently the only Ebola vaccine with demonstrated clinical efficacy in a ring vaccination clinical trial. It has been shown to be reactogenic but immunogenic and safe in several phase 1 clinical trials. Reactogenic just means that it causes some reactions primarily at the site of injection, such as redness and swelling, but immunogenic means that it stimulates a protective immune response. A study was done during the outbreak in the DRC of 2 new monoclonal antibody drugs, MAB114, which was produced from a single human monoclonal antibody derived from an Ebola survivor, and the REGN-EB3, which is a co-formulated mixture of 3 human IG1 monoclonal antibodies. These were compared to ZMapp, which is a triple monoclonal antibody agent that's been used previously in Ebola outbreaks, specifically during the West African outbreak, and then remdesivir, which is a nucleotide analog RNA polymerase inhibitor, which has been used against a number of different viral agents, including currently COVID. So you can see from the graph that the 2 new antibody drugs significantly reduced the incidence of death compared to ZMapp and remdesivir. This is interesting news because it's an indication that the development of monoclonal antibody drugs may be a way forward in coming up with new treatments for viral diseases in general, not just Ebola. So, for instance, think about COVID and President Trump's treatment. Remember, he received remdesivir, which is currently the only drug that's approved by the FDA for treating COVID, although there's data to suggest that it's really not very effective. But he also received an antibody drug treatment. And it's very possibly this antibody drug treatment that he received that is responsible for the remarkably fast recovery that he showed. So this is an indication of an exciting new development in terms of treatments, possible treatments for viral diseases. An important part of dealing with diseases like Ebola is proper PPE, or protective personal equipment. And You can see here the type of equipment that's used by somebody who's dealing with a patient with Ebola. This is very serious. You have to have goggles, scrubs, overalls, aprons, double gloves, boots, and so on. So the kind of personal protective gear that a person would wear is very substantial. However, you can see that they're not wearing a respirator per se because we don't normally think of Ebola being transmitted through aerosols. But it's a very complex, cumbersome kind of activity. So people who are going in to deal with patients have to put on all this protective gear. When they come off, all this protective gear has to be decontaminated, and it's expensive as well as time consuming. So there's been a lot of interest in trying to come up with newer solutions and perhaps more patient friendly solutions for dealing with people. Because, well, let's look at the care. So let's look at an innovative response to ways to isolate patients infected with Ebola. This is the Care Cube devised by other labs. It's a portable, disposable, and low-cost negative pressure unit that one person can inflate in just a few minutes. And this makes it possible to respond quickly and all but eliminates the need for cumbersome hazmat suits. One of the main pillars of any effort to contain and prevent the spread of Ebola, and one that's perhaps often overlooked, is community engagement. To get treated, people have to trust aid workers and the systems in place. Many communities, particularly remote ones, are suspicious of outside help, especially when that help shows up dressed like stormtroopers out of Star Wars and whisks infected loved ones out of sight to quarantine and isolation. The Care Cube reduces the scare factor with an aesthetic that's a lot less Star Wars and a lot more going camping or kids' birthday bouncy house. Transparent windows let loved ones see their family members receiving treatment rather than simply wonder where they've been taken. And there's even a hug wall that allows for physical comfort in the form of hugs and hand holding. Not only does the Care Cube make for a more human experience for families and patients, which will build community trust in the process, but it's also safer and more convenient for medical personnel. Where existing solutions tend to rely on those hazmat suits, those suits have a high potential for user error and thus infection. So this Care Cube inverts the whole model. It focuses on containing the patient and allows for the vast majority of caregiver interactions to happen safely from outside. And the hug wall isn't just for giving hugs. It gives medical personnel protected access to the patient and the entire interior of the cube, making simple medical procedures like checking vitals possible without the need for suit changes. An airlock system with two-stage zippered entrances allow food and other items to be safely passed into the cube, as well as for waste to be safely passed out. A fan circulates fresh air while a membrane-based gravity filter removes the virus before air leaves the tent. And the Care Cube is wholly customizable. Software tools allow the caregivers to tweak containment designs to meet the specific needs of the communities and the geographies that they serve. So you can see on the slide what an individual cube looks like, first of all, and then how a number of these cubes can be put together to provide an area where doctors can be responding to a number of patients. It's really an innovative approach, and the fact that it reduces the need for these hazmat suits makes care much more efficient. Now let's switch over to Marburg virus. Marburg virus disease is a highly virulent disease that causes hemorrhagic fever with a fatality ratio of up to 88%. So it's in the Filoviridae, it's just in a different genus than Ebola virus. Two large outbreaks that occurred simultaneously in Marburg and Frankfurt, Germany, and in Belgrade, Serbia in 1966 led to the initial recognition of the disease. The outbreak was associated with laboratory work using African green monkeys imported from Uganda. Subsequently, outbreaks and sporadic cases have been reported in Angola, the DRC, Kenya, and South Africa. In 2008, 2 independent cases were reported in travelers who visited a cave inhabited by fruit bat colonies in Uganda. Human infection with Marburg virus disease initially results from prolonged exposure to mines or caves inhabited by these bats. Once an individual is infected with the virus, Marburg can be spread through human-to-human transmission by direct contact through broken skin or mucous membranes with the blood, secretions, organs, or other bodily fluids of infected people, with surfaces and materials such as bedding and clothing contaminated with these fluids. Supportive care is all that's available, so rehydration with oral or intravenous fluids and treatment of specific symptoms improves survival. There is no yet proven treatment available for Marburg, but they're developing a wide range of treatments, including immune therapies and drug therapies that are being evaluated. Okay, lastly, we've talked about the medical approaches, the scientific approaches, and so on. But remember, these diseases, these outbreaks affect communities, and it affects not only the patients, but the loved ones. And one of the problems that healthcare workers have faced is how to help the families dealing with the loss of a loved one. Because many of these communities have established burial procedures that may put the family members at risk to exposure to Ebola. So there's been a lot of work at trying to develop safe and dignified burial procedures that will be acceptable to the families. So these are the kinds of things that you might not always think about when you're dealing with an outbreak of a highly contagious virus. But you have to if you want to have successful control of the disease. Now, to finish up this lecture, let's just talk about the shared properties of these 3 virus families. So I put the Rhabdoviridae, the Filoviridae, and the Paramyxoviridae in the same lecture for a reason, because they have a substantial number of shared properties. First off, of course, they're all negative sense single-stranded RNA. They all have helical symmetry of the nucleocapsid. They have to carry a virally encoded polymerase in the virion because, of course, they're negative sense RNA. But the genome transcription is sequential and yields predominantly overlapping subgenomic messenger RNAs. So remember that the transcription of messenger RNA from the full-length minus strand RNA is an attenuated process, and it moves from 3' to 5' end. The replication of all these viruses occurs in the cytoplasm of the cell, and the progeny virions bud at the plasma membrane.